We have developed a defined, reproducible, and long-standing protocol for small RNA sequencing and for analyzing normalized reads using open-source bioinformatics tools.
The main advantages of our strategy are that it accurately collects microRNAs through gel purification, and that the bioinformatics analysis can be applied regardless of how the microRNAs are collected.
High-throughput sequencing of microRNAs can reveal insight into disease mechanisms, the processing of microRNAs, and the accurate quantification of gene therapy approaches that deliver small RNAs.
Make sure to work in an RNase-free environment and to keep all of the RNA products on ice throughout the procedure.
Visualizing the gel cutting steps will help viewers to identify the correct size of the products required for downstream applications.
For 3' adapter ligation, combine 11 microliters of RNA with 1.5 microliters of ATP-free 10xT4 RNA ligase reaction buffer, 1 microliter of polyethylene glycol, and 0.5 microliters of 3' linker. Heat the samples at 95 degrees Celsius on a thermocycler for 30 to 40 seconds, followed by cooling on ice for 1 minute. Add 1 microliter of T4 RNA ligase 2 and incubate the reaction at room temperature for 2 hours. While the samples are incubating, pour a 15% polyacrylamide gel between 0.8 mm separated glass plates in a plastic cast and insert a comb.